Hello everybody, welcome back to Biology Essentials number 1738. I am Mr. Sanding Boy, and today we'll be discussing the humoral immune response. Now, while you may be aware of the cell-mediated immune response, this is entirely different. Unlike the savage, cannibalistic cytotoxic T cells, these cells are a bit more harmless. Nah, I'm just kidding, they still kill pathogens, but they're more intricate. And so, how, you may ask? Well, you'll see. But for now, let's get started with today's lesson. So, what is humoral immune response? Well, you might think it involves laughing and comedy. It actually involves body fluids, hence the humor name. And basically this process occurs inside the blood and lymphatic fluids, and so in doing so, these B cells are actually taking out all the pathogens inside these fluids. So how this process starts is that much similar to a cell-mediated immune response, as both require the T helper cells to kickstart. And so let's go with that. So basically, once a pathogen is found inside the body, an antigen-presenting cell, whether it be a macrophage, a B cell, or a dendritic cell, encapsulates this pathogen and then starts to break it down. And so only its antigens, which are on the outside of the pathogen, are left. And so what happens then is that they rise up to the outside of the antigen-presenting cell, and as a result of this, the MHC then kind of becomes like a carrier, or a presenter, in this case, for the antigen. And so once it's there, basically T helper cells will start coming through and just like trying to bind with it. However, it's only until the specific antigen or antibody is found for the antigen that the T helper cell will bind with the antigen presenting cell, as the antibody that's on the T helper cell has a specific shape that matches a specific antigen. And so once those bind together, basically they lock up the CD4 protein with the T helper cell, ensures that the bind stays intact, and cytokines are then released by both the antigen presenting cell and the T helper cell. And what happens with that is that the cytokines promote proliferation, so cell duplication. And so as a result of that, more T helper cells show up, or are produced, and basically they all have the same specific antigen receptor to the infestating pathogen. And so with that, cytokines are continuously secreted by the binded APC and THC, as well as the new proliferated T helper cells, to be able to summon the cytotoxic T cells, which are used for cell-mediated immunity, but more importantly, the B cells, which are used in humoral immunity. Now let's get on with the fun stuff, shall we? So once the B cells are signaled via the cytokines, there's kind of a issue. The thing is that there's many different kinds of B cells going on. Like, some of them will not be breaking down the pathogen, some of them will, but it's going to be the wrong epitope, which is the shape of the antigen, and then some of them will have the right one. And so basically, it's just kind of shuffling through the pile until, hey, we found the right B cell with the specific epitope. Once it's found, an activated T helper cell, so aka a T helper cell that's been stimulated by cytokines, it'll go to the B cell with the specific antigen that's needed, and will bind to it. And so once it's bound, the B cell is stimulated by the cytokines that the activated T helper cell also sends. See, it's all about cytokine stimulation. The B cell that's stimulated, once again, will proliferate into two different variations of B cells. And these are memory B cells, which will be used in the second humoral response, and plasma cells, which are used in the primary humoral response. So, plasma cells. Basically, these are the active B cells and usually are used in the primary humoral response. Why? Because they're a one-use thing only. And so, essentially, these things release their antibodies onto the pathogen. And as a result of doing that, three things occur. So one of them is neutralization. And so what happens is that because the antibodies bind onto the antigen of the pathogen is kind of stuck. It becomes a null body, and so its ability to infect is inhibited, and so it's kind of like, oh, well, I can't do anything. Oh, rip. And then, and so opsonization is another thing that will occur. And so what happens is that once the antibodies are attached onto the pathogen, phagocytosis is kind of like promoted. And so basically these antibodies are like, hey, you see this pathogen? Kill it. Kill me in the process too. Then again, antibodies don't really have a life, but you get what I mean. And then lastly, we have complement activation. And so complement activation expresses the complement protein inside a pathogen. And in doing so, it enhances opsonization, basically, killing the cell through the pathogen through phagocytosis, as well as it can kill the bacterial cell directly as a result of the protein being expressed. And so, yeah. And lastly, but not least, we have the memory cells. And so essentially these are used in the second humoral response and are the final push to remove these pathogens from the humor in the body. And so basically there's nothing much to be said except do the same thing as plasma cells, but are kind of delayed in response. So like the primary response will be done to take out as much pathogens as it can. If there's some left over, or it's just to eliminate for good from the cell or from the body, the memory cells come through and just take them all out. And so with that, that's humoral response in a nutshell. Thank you all so much for watching, and I hope that was helpful.